I first met Dr. Seth Lederman in the fall of 2020 when I, along with everyone else on the globe covering biotech, was being inundated with pitches to speak with the hundreds of clinical stage biopharma leaders seeking to solve the then new COVID crisis. I've talked with a lot of them since, but the approach at Tonix Pharmaceuticals, where Dr. Lederman serves as co-founder, CEO, and chairman, is unique in a number of ways that we're going to discuss on today's episode of the Business of Biotech. For his part, Dr. Lederman followed up his Princeton undergrad in chemistry with a Columbia MD and stayed there for a long, long while as a teaching academic, directing research in molecular immunology, infectious diseases, and the development of therapeutics for autoimmune diseases. It's on that foundation that he became a biotech entrepreneur, and I'm honored to reconnect with Dr. Lederman. Welcome to the show.
Thank you for having me back on.
It's my pleasure. So, as I noted, I want to start out getting to know you a little bit before we get into the work that Tonix is doing. But as I noted, you spent more than 3 decades at Columbia University. You're deeply rooted in academia. That's clear. But during that time at Columbia, you got a taste for the business side of biotech. You discovered the CD40 ligand, made an antibody for it, and partnered with Biogen to develop it. And from what I gather, you tell me where I'm wrong on this, but from what I gather, that was sort of your first brush with the industry side. And from there, you've migrated to the point where you're a full-time biotech entrepreneur. So I think I framed it up somewhat, accurately there, but why don't you kind of walk us through that experience and how it kind of transpired from your professorship at Columbia to where you are today?
Sure. Well, thank you, Matt. Yeah, I think you nailed it. When I was a professor, my first major pharmaceutical collaboration was with Biogen over the development of a humanized monoclonal antibody against CD40 ligand. And this is an era when monoclonal antibodies were brand new. And as a matter of fact, I was working through a period after Centocor, which was later bought by J&J, had a failed product in septic shock. And actually, there was very little interest in therapeutic monoclonal antibodies for a lot of the time I was working in that area. But maybe I should take a step back and say I went to Princeton, as you mentioned, and Princeton is, at that time was a pharmaceutical university. I wouldn't say a trade school, but certainly we were right in the middle of the pharmaceutical, the world's pharmaceutical industry at that time was centered in New Jersey, and Princeton was in the mix. So I was, from an early age, I also grew up in Manhattan, where many, BMS and Pfizer were headquartered at that time. So I very much grew up in a world of pharmaceuticals and R&D. And I was, as you mentioned, a chemistry major and then went to medical school at Columbia. So I've seen 2 things. I've seen the peak of the New Jersey and Manhattan pharmaceutical industry. And I've also watched the emigration of a lot of these companies to Cambridge and to other places. So that's given me an interesting perspective. Mm-hmm. But I was very much an academic when I was at Columbia. And I think that academics and the pharmaceutical industry occupy very important but different roles in what's really an ecosystem of this community that brings forth new therapies. So I've been pleased to be part of that community my whole career, and it's been exhilarating to be on the biotech side of it now.
Yeah. So tell me, expand a little bit on that, sort of the collaborative spirit between that you saw and in some ways, many ways, fostered in your own career between academia and industry. Like you mentioned that they occupy, a similar space, and it's interesting. I've spent time talking with biotech execs who sort of disparage the gap between academia and industry, and others who just laud the collaborative nature and the value of these relationships. So I think the perspectives are varying out there. I'm just wondering what yours looks like.
Well, I've seen a very interesting development over my career, where when I was, young in college, medical school, and as a professor at Columbia,
Mhm.
there were two poles. There was academia and big pharma, and big pharma at that time was really a remarkable institution. And I think if the world was perfectly run, we probably would continue to have big pharma operating as it did back in those days, because big pharma had phenomenal resources and they had just unbelievable basic science going on, within their walls. And the typical big pharma, I did a mini sabbatical at Merck in 1990. And so a lot of my views on that are of big pharma can be expressed by Merck in those days. And you had, a large complex with a wall around it, but inside the wall, you had chemists interacting with crystallographers, interacting with biologists, interacting with, it was just amazing what was going on there. And academia wasn't nearly as sophisticated. But two things happened, I think. One is that the investors got greedy, and I don't know why the pharmaceutical executives went along with it, but basically big pharma became a very stripped down version of that. And all the basic science was stripped down and cut out. And for whatever reason, there was this idea that big pharma had to be more, financially lean. And that became the new contest, is how to shed core things like R&D, how to shed manufacturing, how to ship things, overseas, all this kind of thing. So, what you see of big pharma today is just I don't know if you'd say the shadow of its former self, but a very skinny down version that's, a profit-making machine, as opposed to when I was doing my mini sabbatical at Merck, the joke that was well regarded by people at the company was it was internally called Merck U. So it's like Merck University.
Mm-hmm.
So I think what happened, with that, transformation of big pharma into this, profit making, machine, is that biotech really played a bigger and bigger role.
Mm-hmm.
So when Merck used to develop drugs, they would do their own clinical operations team, and they would, instead of starting with one phase 3 study, they'd start with 2 or 3 phase 3 studies simultaneously.
Yeah.
And all sorts of things. There's just so many resources around to do a better job, and there's a lot of emphasis on a very orderly progression through dose finding, pharmacodynamics, all this kind of thing. So what's happened now is you have big pharma, for the most part, being a sales and marketing organization and kind of a merchant bank or something for these more advanced projects. But
Mm-hmm.
now you have this burgeoning biotech industry that is so exciting. But biotech, all of the risk has been concentrated in biotech. I guess that was one of the differences. In the old days, the scientists took on less risk. And I think that what's happened now is that the risk has been pushed all on biotech, and to a large extent been pushed down onto the scientists. So it's new, exciting. I mean, I think critics of the old big pharma would say that they weren't good at innovation. I'm not sure I'd agree with that, but biotech certainly is a dynamic culture where there's a lot going on, and a high premium on innovation, and dynamic organization and dynamic research interests.
Yeah. Yeah. When was it that you sort of decided to concertedly make the transition to biotech? I mean, there's obviously there's overlap there. I mean, you maintained associate professorship for some time, I'm sure, but give us a sense for when you sort of made the hard left turn into biotech.
Yeah, or the hard right turn.
Right turn, right.
which direction it was.
That's
But I was a tenured professor at Columbia University, so I had a lot of freedom, at least for a day a week. And also part of my official duties was working with Biogen on the CD40 ligand project. So I got a real education. One of the fun things about collaborating with Biogen at the time, it was the premier biotech company. And it was also one of the early residents of Cambridge and Kendall Square. So going to visit them and having meetings was fun, but it was also, Biogen was a very prominent presence in Kendall Square. Now, when I go to Kendall Square, it's hard to find Biogen because there's so many buildings that have gone up between, the Red Line stop and everything from, where it used to be in the hotel we'd stay at. Anyway, so when did I decide? I think that what I realized in the Biogen thing was I was a leader in molecular immunology, and I discovered the CD40 ligand, as you mentioned. But when I was working with Biogen, I realized that one of the key functionalities that even Biogen could improve on was clinical development. And I am a physician. I was a professor, associate professor of medicine, and I worked for many years in the division of rheumatology and practiced rheumatology, taught rheumatology. I realized that combination was very valuable in industry. And at that time also, the university was going away from a model where people were what was called triple threats. I was trained to be a triple threat. A triple threat meant research, clinical, and education. And I was very involved in all three of those areas. But increasingly, universities were pigeonholing people into just basic science, just clinical, or just education, as those became somewhat more specialized. So I found that the environment at Biogen, where there was a lot of interaction between clinical development and R&D, was very stimulating. And I felt that my expertise in clinical medicine, as well as my basic science training and accomplishments, were better suited to biotech. And also, I felt I filled a bigger need. And there were a lot of smart people doing molecular immunology, molecular virology, genomics, the things I was involved with there. So while I was fortunate to have been a leader in some of those areas, at least from time to time, I felt that in biotech, I was more needed and potentially more valuable.
Yeah, the potential to help more people and have a broader impact, and that you have. I mean, I already, and you're working aggressively toward it, because it kind of gets to one of the topics I want to talk about, and that's the breadth of your pipeline, breadth and depth of your pipeline at Tonix, and we'll get to that in a minute. But if I get to that too quickly, we won't sort of get the genesis story of Tonix. So you co-founded the company in 2014. What was your, I guess, motivation for launching your own? Like you as you said, you made up your mind to move into industry. You could have done that in a lot of different capacities, right? With your credentials and your experience, you could have moved into probably pretty much any company you wanted to. You could have moved into early biotech as a chief science officer, I'm sure, any number of roles. Why co-found your own?
Well, thanks. That's an interesting question. And I'm sure, like many other people, my life has been a series of stochastic events, opportunities, different things that happen. I'm not sure I can draw a straight line, but I will say my career in biotech has been exclusively working on companies that I've founded and ideas that are important to me. And to jump ahead to the broad pipeline we have, which now includes a number of things that we've brought in through M&A,
Mm-hmm.
partnering, collaborations.
Yeah,
So
like
it's no
20
longer,
some candidates.
yeah, yeah, we have
Am I in the right
might
ballpark
have
there?
something like that. But
Yeah.
the one theme that has stayed the same is our criteria for bringing something into Tonix is that we have to basically be in love with it. It
Mm-hmm.
just has to be something that I or the people on the team working on it are passionate about. Because if you're not passionate about an idea in biotech, it's just too hard. No one can succeed in biotech unless there's at least one person who is passionate about a drug. So that is the common theme of everything. And in the beginning, when I, moved out and, founded first one, then another, then a company, it was because these were ideas that I really felt needed to be done. And I felt that in order for them to get done, I had to play a role.
Yeah. So that's it's an interesting point. And I've often wondered this. I want to ask you a question about the passion and the love, the commitment to a candidate or an idea in this business. And I've asked other people this question, but it's one of those questions you give variable answers to. You've also got to know when to fail fast, fail cheap, whatever you want to call it, cut bait and run. Pick your poor analogy or metaphor. What do you do? Like when you have a situation where perhaps you've acquired something at a too early a stage that doesn't pan out the way you wanted it to, or you're working on development yourself and you've got a team or a person, deeply rooted in that project and cheerleading for it. And from a business perspective, it's just not going to work out. You just need to let that go. Like, is that a tough spot for a CEO to be in?
I don't know if it's a tough spot. It is the role of the CEO and to make those decisions and make those decisions with a team. And it's true that sometimes when you are making these decisions, it requires some convincing. So it's very difficult. People do get overly involved with things. And then when we make decisions, we have to make them and move on. But, clearly it's easy if there becomes a signal of patient risk or, something that, involves safety. Those are easy decisions. Yeah. But there are hard decisions, particularly after, in later stage development, if a trial misses and, you can look and see better, ways to have done it. There is a question about when are you still learning and, when are you just spinning your wheels? So, these are hard decisions to make. But I will say that, many successful drugs have, I think almost all successful drugs have had stories where at times during their development, not everything looked positive. Prozac is one of the biggest successes of all pharmaceutical history. And I believe there were more than 5 studies that were done to get 2 positive studies. So I think that sometimes, you have to understand the disease you're targeting, condition you're targeting, the complexities of studying it, and to continue learning through the trial process.
Yeah. When you were talking about the good old days of big pharma, and you talked about, running 3, your eyes lit up with a little bit of envy when you talked about running 3 concurrent trials. Like, you wish that you had the resources, you were resourced enough to do that. Any emerging clinical stage biotech company CEO would. You're pretty aggressive though, like from like I said, from a breadth and depth of the pipeline. Not only do you have 20 some candidates, but they're across modalities, across disease areas, spanning CNS, immunology, infectious disease, and rare disease even. So that all that leads to the question. I want to have a conversation around how you mentioned acquisition. I'm sure there's been some internal development. How have you managed to develop that deep pipeline so quickly. I mean, it's relatively quickly. I've talked to plenty of companies that were founded well before 2014 that either haven't been able to or haven't chosen to get that aggressive with the development of their pipeline. So let's just talk through that. How'd you get that set up?
Sure.
And I, Dr. Lederman, I don't, I'm not asking you to take me, on a room by room tour of each candidate. I just want to get a sense for what the strategy's been.
Sure. I think it's a great question. I think it's, being a CEO of a public company, it's, talk about an ecosystem, we are involved with our investors and we're listening to our investors and trying to do things that make sense for our investors. So I think that public companies generally have a lifespan and a cycle of life. In the early days, when they are dominated by VCs, they tend to be rifle shots, meaning one program, one platform, one idea. Mm-hmm. And that is, that's a theme of most VC-backed companies. And I've worked very productively with VCs in the early stages of my biotech career. And I think VCs are now an extremely important part of the ecosystem. But from the VC perspective, they like to focus each bet on one idea, either a drug or a platform, and they diversify their investment portfolio by doing multiple investments. So VC fund might have 10 programs and they're hoping to hit on 2 or 3, hoping to break even on 4, and then to, they expect to lose money, whatever. But their diversity is across the portfolio. So that's a private company model. A public company model, at least for our investors, is one where the investors appreciate a diversification of the risk across more than one project. So it's a very different model for us as as you say, a public company has been public for a while. We provide investors diversity across the across a portfolio. But if you think about it, we're not doing an apples to apples comparison, because VCs answer to their limited partners, and the limited partners, who are the real money behind these funds, want diversity. So the limited partners want diversity. The VCs give it to them by making multiple bets in each fund. We answer directly to our investors, who are directly investors in Tonix. So we don't have a middleman between us and our investors. Mm-hmm. To use, I have great friends who are VCs, and they work very hard at what they do, and they have tremendous expertise. But I'm just saying in this context that there's no intermediary between us and our investors. So our portfolio gives our investors diversification of risk over these different opportunities.
Yeah. Sound strategy that's well understood. Give us some insight into how you manage it, though, because effectively, if you look at each one of those VC backed startups who has a single perhaps molecular entity in the rifle shot approach, you're effectively CEO of 20 of those companies with the pipeline that you have now. Obviously that's a bit of hyperbole, but you get the point. Like, you've got a management and a staffing and an IP management challenge when you've got everything from rare disease candidates in the ATMP space to vaccines to small molecules. So how do you go about that? Do you do a lot of outsourcing? Do you you have an incredibly diverse staff? What does that look like?
Great question. So we, again, talking about the ecosystem model, we are specialized for the most part in the transition of drugs from preclinical through early clinical to phase 2. So that is our bread and butter. And we have a team that has functionalities that are specific to that process. For example, we have a great group that does manufacturing development. this is something that universities don't think about, don't do. When we get things from universities, we have to pick it up, make, the compounds at GMP, formulate them, do PK, all this kind of stuff. So I so we have a multifunctional team that focuses on those areas, and we specialize in those activities, and we're very good at it. And that's why there's a commonality between a lot of these different programs and what our expertise is. The programs come in on one end, and we do those activities that we're good at, file INDs, get approval to do human studies, and take them along the way. So I think that it's probably best to think of us as this part of an ecosystem where we're going into the clinic and advancing things, into phase 2. I think phase 2 is the key place that we play. And one of the other things that I should mention about our role with VCs is, they are also very limited in the time frame with which they can pursue investments, because of a typical VC fund has a finite life period. And they have to invest the capital and get returns over a certain period of time. So sometimes when programs take longer than they had expected, they really fall out of the opportunity. Many VCs can't reinvest in the same project, or they can't, if it's in fund 4, they can't invest in that from fund 5. So there are a lot of kind of opportunities that have come up for us where a lot of time and money has gone into a project. It's very interesting, but it no longer fits into this, very specific kind of role that VCs play. So because we are, some people call a public company evergreen, right? That we have capital and activities that are not limited by the the confines or the agreements of a closed-end VC fund that's time limited. Yeah. That we can take things that have promise, have gotten a little bit beat up, but need some help and need to get further along. So again, part of the ecosystem. So that's why I think it may look more complex than it is. Because when we're bringing in something, as I said, the relevant people in our team have to love it, and that also means that they have to have the bandwidth to do it. Yep. Now, you asked about outsourcing. Yes, we do a lot of outsourcing until relatively recently, even going back to 2018. You might have even called us a virtual company. But over time, we've expanded our clinical ops team first. And now over time, particularly in the area of infectious disease, we have significant internal capabilities, including bricks and mortar, and trained people and other things that they're doing. So we've evolved over time. But I think that we're also, as I said in the beginning, about how much of it is pursuing a vision like this and how much of it is just reacting to times. I think that what you're seeing now, particularly with the pandemic, is that the global supply chains are a mess. Yeah. And what had become, the outsourced research model depended a lot on other countries. As you probably know, APIs are made in India by and large. A lot of contract research, is going on in China. And, before the pandemic, it was great to operate in that kind of a virtual model. But travel has become harder. Importation has been harder. Just sending things across borders is harder. And there's a growing recognition that the United States needs internal resources, particularly for areas like biodefense, infectious disease, areas where we're very active. So to some extent, we're reacting to the times by increasing our internal capabilities, our bricks and mortar, and these other capabilities. So it's To be in biotech, you have to be dynamic. You have to be versatile. You have to adapt to the time. And
Yep.
I think that we're doing a good job of staying with the times and also staying ahead.
When you're striving to excel in a new arena, the best guides are the ones already doing it well. The Business of Biotech brings those voices forward to help new and emerging biopharmas turn their innovations, like mRNA and cell and gene therapies, into clinical realities. Tune in and subscribe for insights on hiring, regulatory, and other need-to-know topics for biopharma leaders. The podcast is brought to you in collaboration with Cytiva. Check out their resources at cytiva.com/emerging biotech. That's CYTIVA.com/emerging biotech. Perfect. Is that brick and mortar space that you've invested in, like lab space, or are you actually building or considering building manufacturing capacity of your own?
Both.
Okay.
Last year, on October 1st, we closed on the acquisition of a 48,000 square foot R&D facility in Frederick, Maryland. It was built as BSL-3, but it had been operating at BSL-2. We have taken the steps to upgrade it back to BSL-3, but we're waiting for an inspection, some other, certification to get back at BSL-3. So this is Frederick, Maryland. If you're familiar with that area, is at the north end of the 270 corridor. So, Frederick is at the top, and then it goes progressively down Gaithersburg, Rockville, Bethesda, et cetera. So we're at the top of that. We're just a few miles away from Fort Detrick, which is the home of USAM RID, the leading infectious disease institute of the Department of Defense, and also a major NIAID facility at Fort Detrick. So there's a lot of cross-fertilization, and both in terms of personnel, we've hired some great people from that campus, but we also have burgeoning collaborations with them. So we have really phenomenal capabilities in this facility that's fully operational and running now.
Wow.
And then in just south of Boston, in New Bedford, we are about to open a manufacturing facility. It's a manufacturing process facility that can do launch scale manufacturing, but is really focused on phase 2 and phase 3 biologicals, and particularly biologicals relating to our vaccine program. So again, those are capabilities that if we thought that we could contract out for those activities, we probably would have continued to contract out. But when we realized that we couldn't be second in line, we had to control our destiny, we needed some specific, tooling, machines, bioreactors, systems, that it made sense for us. And together with our investors, we, have been able to build this really phenomenal capability that we expect will be, open by the summer.
Well, that's excellent. Congratulations. And it makes sense, in my mind anyway, it makes sense to do that if you can afford to, because given the capital crunch these days, given the, I guess more discerning attitude of the biotech capital markets and venture capitalists around what they're offering startup biopharmas, I don't anticipate that we'll see a whole lot of early stage manufacturing capacity growth, which in my mind, I think would actually put more pressure on the outsourced community, which is to your point already asking people to take a number and wait. So congratulations on that. It sounds like a strategic and well-planned move.
Well, fortunately, we were ahead of the curve, but I would say there's a disconnect right now between United States policy and what's happening on the ground. I think that there were a lot of policy statements saying that the United States needed domestic manufacturing, they needed domestic R&D, a lot of other things. Yeah. And I think that in 2020 and 2021, you could accomplish a lot because the biotech markets were open. So I am pleased that together with our board and investors, we were able to develop these capabilities when it was possible to do it. I do think that once some of the politics blows over, I don't know, I'm not an expert in politics, but I do think that there's going to need to be a huge commitment of the federal government to jumpstart some of this re-domestication of pharmaceutical manufacturing. You see a lot of it happening in the chip space. President Biden, the State of the Union, highlighted the things that Intel is doing. And there's an act that is specifically being floated around to, rebuild American competitiveness in chip-related technologies. And I foresee that the same thing has got to happen in the biotech space. So I think that we're in a very good position to benefit from that because we have the expertise of operating and even building complex manufacturing. So I think that, we are very well positioned to benefit when the government gets around to doing it. And they will get around to doing it. Our government doesn't always seem like it operates in a straight line, but, I think over history, we've gotten, most things right. So I think that this will be an important advance. And as I said, we're very well positioned to be part of that.
Excellent. I want to move on and start talking specifically about your COVID program because I'm looking at the clock here, Dr. Lederman, and I feel like we should have set 2 hours aside because we've got a lot to cover here. And I'm really appreciating your responses to my questions, the depth and detail. But I'm only halfway through my question set, so we need to move on. And I want to get some perspective on your COVID strategy. You've got several COVID related candidates in the pipeline. I want to start with the vaccine. And I want to just throw some numbers out there and get your take on them so that I can better understand what that market opportunity looks like. So there's a count out there being tallied by the Regulatory Affairs Professional Society. It is not comprehensive, but it's a count of nearly 100 clinical COVID vaccine candidates globally. They're kind of keeping track of clinical stage when one drops off and another adds on. As I said, I think it's 90. It was 96 when I checked yesterday. Again, not comprehensive. There are a couple hundred more, that we could identify through various sources in preclinical stages. So this COVID vaccine field is a crowded one, right? Like, and for understandable reasons. 18 months ago, 2 years ago, everybody wanted to jump on because everybody wanted to capitalize on the opportunity and be the hero and do the right thing for humanity. We've seen several approvals globally now. I don't know, 15. I mean, we're familiar with the ones we are here in the States, but I think there are like 15 approved COVID vaccines globally. So last April, Tonix and Kansas State, if I'm not mistaken, announced collaboration to develop a vaccine candidate for COVID-19, prevent COVID-19, utilizing a novel live virus vaccine vector platform. So there's a number of questions in there. Why get into the game? Because like I said, from a simplistic perspective, like mine, it looks like a crowded field to jump into. Two, it's everybody knows that RNA, siRNA, mRNA, anything with RNA in it is super sexy right now. So why a novel live virus vaccine vector platform?
Great. Well, thanks for that question. So first of all,
a lot of words for that question, right, Dr. Lederman? But you get where I'm going.
I started my career in the AIDS era when I was a third-year medical student. The New England Journal of Medicine article came out about a new disease in gay men in San Francisco. So the first 10 years of my career were devoted to AIDS, and I did significant work in that area. And like many other people now, including Dr. Anthony Fauci, who obviously rose to fame in that period. I'm younger than Dr. Fauci, but did not have the same impact on HIV as he had. But I think that the reason we're in it is because we think we have something very unique and important to bring to the game. There are, as you mentioned, over 100, some people say 300 candidates in development. And from my perspective, there should be more. I mean, this is something that shut the United States down for 2 years. Uh, Former Treasury Secretary Larry Summers called this the $16 trillion pandemic.
Mm.
And we're not out of it yet. So I don't know how many programs there should be or how much we should be spending, but I don't think there are enough programs, and I don't think we're spending enough. Certainly government is not spending enough, in my opinion.
Yeah.
But so we have a very unique take on it, and that relates to live virus vaccines.
Mm-hmm.
Live virus vaccines were the first vaccine, literally Edward Jenner in 1796, so 220 years ago, did the first vaccine. He used horsepox to vaccinate people against smallpox. That turned out to be the most successful vaccine ever. We have picked up on that, and we've been working on vaccines since before COVID. So we had already been working on our internal horsepox program. That the smallpox vaccine form is called 801, and our COVID vaccine programs are called TNX 1800, 1840, and 1850. So basically, we used a lot of new technology to recreate what we think is very close to what Edward Jenner used. So actually, we used synthetic biology to create it. When we did it, it was only the fourth virus ever synthesized. I believe it's still the largest virus ever synthesized. But we did it because so much was known about this vector. It had eradicated smallpox in Europe. It has really great features in that it brings the antigen to the right place. It gets into the cells express it, and you get strong immune responses. And by the way, smallpox was a respiratory pathogen. And this shot in the arm eradicated smallpox. So we think that this is a very important potential way. It's relatively inexpensive to manufacture. It has much better handling characteristics, so it can be used in places that don't have good refrigeration, poor supply chain, the rest of it. So there are many reasons to work on it. But now let me go into the reason about who needs another. The mRNA vaccines were a remarkable advance. I believe that Drew Weissman, Katalin Karikó, and Peter Cullis, the people that were involved in this work, in my opinion, deserve Nobel Prizes. The mRNA vaccines have given us breathing room, but they are not the end of the problem. They have two major limitations. One is the short duration of protection. Short duration means that you have to get boosted every 4 to 6 months. And as the head of FDA's vaccine branch, Peter Marks, said last week at the Vaccine Advisory Committee, that's not a tenable or sustainable program. The second limitation of the mRNA vaccines is that they don't block forward transmission. Forward transmission means if I am infected and vaccinated, will I transmit it to you? Unfortunately for Omicron and Delta, it appears that vaccinated people are pretty good at continuing to spread it. So in my view, in order to really get a handle on this, we need vaccines that provide durable protection and block forward transmission. So if you look at the history of vaccines, smallpox, MMR, measles, mumps, rubella, those are 3 live virus vaccines. The early chickenpox vaccines, other things, Sabin oral polio. The effective, cost-effective vaccines that have blocked forward transmission historically have been live virus vaccines. Now, this was well known to Moncef Slaoui, who was the first head of Operation Warp Speed. And, if you're handing out Nobel Prizes, and by the way, no one is asking me who should get a Nobel Prize, but I think that Moncef Slaoui would be up there for some kind of a peace prize if you don't want to give him a Nobel Prize. But he did a wonderful job of stewarding the resources to come up quickly with a vaccine. But Moncef Slaoui also said there are 4 types of vaccines that should be evaluated. RNA, subunit, replication incompetent, and live virus vaccines. he got to the first three, didn't get to the live virus vaccines. At that time, Merck, who at the time was a leader in vaccine technology, if you go back to 2020, Merck had developed more innovative vaccines than anyone else. And they chose to work on two live virus vaccine platforms as their solution to COVID. But it never got picked up by Warp Speed. And when it looked like the mRNA vaccines were doing well, Merck discontinued both programs. So we are picking up the mantle of this fourth pillar, Moncef Slaouy's fourth pillar of vaccine technologies, live virus vaccine technology. And we have, as you noted, two different strategies. We have the horsepox that is more homegrown, although it really came from a very productive collaboration with University of Alberta in Canada, which is a phenomenal vaccine and virology institute with a Nobel laureate from Hep C. Not that we're directly collaborating with, but in that department. Yeah. And also Kansas State, which is also a worldwide leading place where we have a second candidate, just like Merck. I mean, it's obviously not appropriate for the head of a little company to say we're just like Merck, but with respect to taking two bets on live virus vaccine candidates, different live virus vaccine candidates to bring the COVID antigen to the body, that's why we're doing it. Let me just give you, let me revert to being a professor for one instant.
Sure.
Every vaccine has two essential elements. One is the antigen. That's the feature of COVID that it's going to bring to the body. That's what's recognized. The second essential thing for any vaccine is a danger signal. And the danger signal is the thing that wakes up the body's immune system so that it mounts a durable response to the pathogen. And while I believe, as I said, the mRNA vaccines are remarkable, they've given us breathing room, they've done a phenomenal job as a first vaccine, I think it's time where we need vaccines that provide more durable immunity and block forward transmission. And I would say that the limitations of mRNA vaccines appear to me to be that they provide an insufficient danger signal. Mm-hmm. Now, in a subunit vaccine, the danger signal is provided by what's called an adjuvant. For example, the Novavax vaccine looks very interesting. They have a proprietary adjuvant. Glaxo has some very interesting adjuvants, actually, that are made in Hamilton, Montana, and they're part of their partnership with Sanofi on a COVID vaccine. But live virus vaccines have a very strong property of providing that danger signal. And that's what we think is so unique about them, that if you put a live virus vaccine into the arm, it activates many parts of the immune system that are related to danger signals, things like toll-like receptors and other warning systems for the body. So we think that that's the important thing to harness on next generation vaccines. Now, before I leave, let me say this point. Unfortunately, we are going to have until the end of time to come up with the best vaccines. Now, Tonix doesn't have till the end of time, but I think we're a very long way away from having a vaccine that is going to bring us, and I mean us, humanity, mm-hmm. into some kind of an equilibrium with this virus that we can truly call endemic. Mm-hmm. It's hard to call something endemic when just in this year, in 2022 alone, 150,000 Americans have died. Mm-hmm. It hardly seems to me like we're on the other side of this pandemic.
Yeah. Yeah. When did you, there's a, risk analysis that goes on. I said that too simply, a risk analysis. There are a million risk analyses that go on when you're determining candidates to develop, to add to your pipeline, to acquire, whatever that might be. In this case, one of them is the competitive landscape. And I'm curious about what you knew about the durability or assumed or sort of foresaw about the durability and ability to block forward transmission of the current mRNA vaccine candidates, in order to have confidence that you might be able to do it better. Because if I remember correctly, there was a good long period of time when there weren't any necessarily straight answers nor expectations around durability or the ability to block forward transmission among the vaccines that have been approved to date. So I'm just curious, like when I think about that paradigm, like that situation, and I think, you had to make a decision at some point while these approved vaccines were already out there in the wild, about whether or not you could do it better. So it had to come at a point where you knew that these guys weren't able to meet those objectives very well.
Well, thank you. First of all, I was hoping they would work.
Of course.
So I was rooting for them to work. But I have been, in this ecosystem for 30 years, more, 40 years if you go back to graduating college. And, I've seen technologies come and go. I've seen things that look great early, not pan out. And this is clearly a brand new technology, the mRNA vaccines. Again, they're remarkable. I've been vaccinated with Pfizer 3 times, and I believe that, if we could vaccinate more people, it would be helpful. But I do think that it was premature to make decisions based on 3-month data.
Mm-hmm.
That's just not the time course for which you normally make a decision about which vaccine you're going to base the national policy on. There are natural time things in the human body. The immune system has 2 major components, the antibody part and the T cell part. And antibodies are known to wane by 6 months, and T cells are known to have more durable years, decades of protection. So the very strong antibody responses to the mRNA vaccines were to some extent predictable, maybe better than expected. But on the other hand, I think that 3-month data, in retrospect, could be called a sugar high. That now that that it's clear that without further boosts, the 1-year activity of mRNA vaccines would not have been a basis to make a national vaccine policy on. And I think that now more and more you're seeing statements out of Sanofi and Glaxo and J&J saying, wait a second, maybe on this very early data, we came up a little bit short relative to mRNA, but, look down the line and, they're either comparable or let's see.
Mm-hmm.
So I think that, one of the personal or human characteristics you need to be in pharmaceuticals is you need to have a long, a long vision, and you need to have the ability to pursue something and to get results over a long period of time.
Yeah.
So and that's one of the features, I think, why public companies ultimately are better suited to develop pharmaceuticals, because they have a so-called permanent capital base, and they can look further into the future, whereas the venture capital model is really predicated on relatively quick returns, which is based on exiting, either becoming public or selling to a public company one way or another. But anyway, so I think I've seen a lot of vaccine technologies come and go. I mean, I saw DNA was going to be the answer to everything. And mRNA was surprising on the upside. I think, it had not really worked for Zika. And, there were equivocal results and other things. So I think they surprised everyone. I think I even saw Moncef Slaoui remark that he was pleasantly surprised that they happened to work so well against this particular virus.
Mm-hmm.
But I think that was early in the game. And now that we see this virus's enormous potential for mutation, immune evasion, and other wily things that it does, recombination, we just have to continue to develop better and better tools if we're going to establish anything like the world as we knew it in 2019.
Yeah. Yeah. Good. Do you have time for two more quick questions?
Sure.
Good with that. Okay. Yeah, I know we're stretching the limits of my my normal time allocation, but I just two more quick questions for you. You alluded to this, so you should be able to cover this pretty quick. You alluded to it when you were talking about the decision to go with a novel live virus vaccine vector platform. So if you look at the vaccine market on the whole, and you guys are working on multiple vaccine candidates, it's projected to be $150 billion market by 2026. Analyst companies, when they look at that number, they say, okay, well, what might thwart that? What might stand in the way of that kind of growth that we're projecting? And the two things in the vaccine market that they point to are purchasing power in developing countries and the huge capital investment required for developing vaccines from the outset. So one, would you agree that there are two threats to the growth and therefore democratization and distribution of good vaccine candidates? And two, if you do agree, what would you consider the response to those two challenges?
Well, our role, again, turning to the concept that we live in an ecosystem, is that our goal is to create and test a valuable technology. And, to the extent that we can bring it forward and validate that it has, that it's useful for human health, that's our challenge. And one of the things I like about the pharmaceutical ecosystem is that if we get it to a certain point and establish these important characteristics, in my view, we will find partners, a partner that would be able to take it to this next level. I mean, look at what BioNTech did with Pfizer. I mean, that was a very smart deal for them. They realized the magnitude of the problem and what they could do with their technology, and they found a willing partner, a great partner, who could put together all of the resources of Pfizer. And by the way, big pharma CEOs usually can be graded in they all do about the same. It's kind of like the tide in a harbor. But in this case, I mean, Pfizer's CEO has just set himself apart from all other CEOs and what he's done in the pandemic. So the recognition that he had to, Albert Bourla, what he did to rapidly move forward, jumped on it, move this forward. It's now the leading candidate. It's the biggest product ever in the history of the world. And it all is, Pfizer's management and what Pfizer is able to do. So a huge accomplishment. But, again, J&J, huge accomplishment. I mean, they had bought some very interesting technology years ago and developed that to a point where they were ready for it. And they've made a huge commitment around the world. It maybe doesn't get as much play in the United States, but for a lot of the characteristics for which J&J was developing it, and they had a really visionary leader, Paul Stoffels, who's always had a commitment to the whole world, the developing world, everything. And, that's also a remarkable story of pharmaceuticals. So I think that, we see our role as bringing forward this technology, bringing it up to a level, showing that it would have these advantages. And as we talked about in the, deciding based on the results of data, which of our live virus vaccines is a better platform, whether they will have these properties and the rest of it. And if they have the properties that we hope we will have, I am confident that we would find, a partner that would recognize how important this is to the world.
Yeah, excellent. Good response. So final question is just a quick snapshot of, as I mentioned from the outset, your vaccine candidate is one part of a suite, if you will, of COVID and SARS-CoV-2 programs at Tonix. So just a quick snapshot of how these things work together and why, what the strategy is from the CEO's perspective. So Tonix is working on this small molecule candidate for long COVID, a COVID antiviral, a SARS-CoV-2 T-cell immunity diagnostic candidate, that is, as well as this vaccine. Is there a collective strategy among these four related by disease candidates?
Sure. And I think it really relates to expertise. I think a lot of people outside the field think scientists are big thinkers and they just imagine great solutions and things come to them. But from my experience being a practicing scientist for 40 years, whatever it is now, I think it all starts with the details. And when you climb into it, and we have so many different specific expertise and reagent advantages and techniques and equipment and the rest of it, that the reason we have so much going on in COVID is because we know so much about it at Tonix. We are not just a casual player in this. As I said, we have a 48,000 square foot facility in Frederick, Maryland, and we're building another one. And we just have phenomenal talent in our scientific group. So I think that there's an efficiency in terms of the reagents, in terms of the assays, the animal models, the clinical development. There's so many ways in which these things may look different, but they actually cross-fertilize or actually related to each other.
Yeah.
So I think that we're much more of a I guess you would call it these programs in COVID have been built from the bottom up, I guess you would say it, you would call it, in terms of the expertise and the human capital, reagents, equipment, facilities, the rest of it that we've developed. And now, there's increased interest in many government agents about pandemic preparedness generally.
Mm-hmm.
And, we are still very focused on COVID. Philadelphia, I read this morning, just reinstated the mask mandate. This new BA2 variant of Omicron is spreading very rapidly in the United States. You may have seen that Speaker Pelosi, Senator Collins, Defense Secretary Austin, White House spokesman Psaki. I mean, this is, we're not out of this yet. So we're very focused on COVID. But in addition to that, the systems and the expertise we're developing are applicable to many other potential pandemics. But we think that with all of the systems we've got with COVID, we can use that as a model system to the extent that people want to think about technologies for future pandemics.
Yeah, that is very important. We hosted a panel, a Bioprocess Online live panel event not too long ago on pandemic preparedness with a couple of great biotech CEOs who are keenly aware of that challenge as well. What advice would you give to others who are in a position like you were, I don't know, back in 2014 when you were launching Tonix, co-founder, getting started, looking back on that span of time that's happened since then? I don't know. It's a great, big, giant question, but what would be like a good, solid, sage piece of advice you would give someone in your shoes back then?
Well, first of all, I think that to be value added in biotech, it's very important to have technical training, knowledge, and a lot of experience under your belt. So my experience in academia, although they may seem different, as we said, academia is kind of a different pole of this ecosystem, but all of my success I attribute to my training and experiences in academia to start with. So I don't know when the right point is for different people to make the move, but biotech is, I think it's fair to say, more dynamic. applies to a wider range of, lets you potentially affect a larger number of people by this, direct involvement with developing drugs and therapeutics.
Mm-hmm.
So I think that for young people making the move, or middle-aged people, whoever's making the move, I think everyone's story is different, everyone's expertise is different, and the opportunities they're presented with are different. The biotech market is a little bit challenging right now. There are a number of companies doing, layoffs, downsizing, things like that. So it may not be as easy to move into it right now, but everything is cyclical. So I would just say, wait for the next cycle. But the other thing is some people have commented that some of the greatest companies have been built in bad markets. So I think that there's probably a lot going on that's going to be, we'll look back in 10 years and say, these were companies that people weren't focused on in 2022, but look at them now.
Dr. Lederman, thank you for being so generous with your time and so generous with your insight, sharing so much with us. I appreciate it. It's been a privilege to spend this time with you, and I think it's some very valuable content you shared with us.
Thank you, Matthew, and thank you for having me on.
My pleasure. So that's Tonix Pharmaceuticals, Dr. Seth Lederman. I'm Matt Pillar, and this is the Business of Biotech. If you enjoyed today's show, drop us a review and make sure to subscribe on Apple, Spotify, Google, or wherever you get your podcasts. We're produced by Bioprocess Online, and we're grateful for our partnership with Cytiva. Bookmark bioprocessonline.com and cytiva.com/emerging biotech for continually updated insight created by the leaders of emerging bio for the leaders of emerging bio. Check those out. Stay tuned for next week's episode. And as always, thanks for listening.